Okay, my name is Warren John, and today I will be talking to you about part of the research that goes on here at the HZDR, which is located in the beautiful city of Dresden, Germany. The topic of the research is the influence of uranium speciation on its transfer to plants. So, we are focusing in particular on uranium as a naturally occurring radionuclide. And we know that plants do encounter uranium in the rhizosphere or in the soil. Uranium is usually never alone. It's always surrounded by various biological and microbial entities, which determine its speciation in the soil, or basically in what form it exists. The speciation of uranium also then determines how the metal interacts with the plant. And by interaction here, I mean both the passive absorption of the metal onto cell surfaces, as well as the active uptake of the metal into the plant. We incorporate both of these phenomenon in a term called bioassociation. Once the uranium interacts with the plant, of course, the plant then responds to the uranium, and there are many ways in which it does this, but probably the most important one today is that of the secretion of metabolites, such as organic acids and chelators. And some of these metabolites tend to form complexes with the uranium, and they could either increase or decrease the uptake of the uranium into the plant. Other responses also include the translocation of uranium from sensitive root tissue into other parts of the plant, and even maybe vacuolar sequestration, where the uranium is then rendered non-toxic to the plant. So this is our field of research, and this is what we want to dive into, let's say. And we will be working with one particular plant as a model organism, and in particular, we're using Brassica napus, more commonly known as canola or rapeseed. And we'll be working with hydroponic cultures of this plant. So first of all, we would like to investigate the change in the uranium speciation in the hydroponic solution, or the growth medium, let's say, as well as inside the plant tissue over time. So how the forms of uranium change. And this would be investigated using luminescence spectroscopy, as well as with thermodynamic modeling. Secondly, we also want to see how uranium influences the type of root exudates, or what the plant is secreting. And this would be checked with liquid chromatography and mass spectrometry. Thirdly, we would also like to visualize or to see the influence of the plant metabolites on the speciation of uranium, and also therefore its transfer and translocation into the plant. This will be done with luminescence spectroscopy and mass spectrometry. And finally, and I would say more ambitiously, we would also like to see the distribution of uranium species in the plant, which we hope to do with luminescence spectroscopy. So as you can see, TR-LFS is going to be the main workhorse of the project. I would also just like to present some of the initial results we got. So we've been working on this for a month now. And the hypothesis we're working with right now is that
You have 1 minute left, Warren.
Thank you. Is that organic acids in particular are able to resolubilize uranium phosphate complexes in hydroponic solution. So we've taken out the plant for the moment, and we're just focusing on the organic acids to see the speciation of uranium in phosphate sufficient and deficient hydroponic growth medium in the presence of these two organic acids, citric and malic acid. Our thermodynamic modeling results predict that with high concentrations of citric acid that we might see a resolubilization of these precipitates of uranyl phosphate, which actually might not be the case with malic acid. And these are the predictions that we have so far. And right now, we're conducting experimental studies with TR-LFS and mass spectrometry in order to verify this. So hopefully we would be able to show you some interesting results in the future, but I would like to stop there for today and thank you for your attention and gladly welcome your questions.
Okay, thank you very much. Great. Questions?